Laser Pulse is brought to you by BW Tech Medical, a leader in light-based devices for over 30 years. Whether you're a researcher or a manufacturer, we help turn ideas into reality. Learn more at www.bwtechmed.com.
I'd like to welcome everybody to this episode of Laser Pulse, a discussion with thought leaders about where PBM and light in medicine is going, where it is currently and where it's going to go in the future. This podcast is presented by AIMLA, the American Institute of Medical Laser Applications, an online platform that's for over 10 years has promoted education to use lasers safely and effectively within a variety of medical specialties. We're joined today by my co-host, Dr. Brian Pryor, who is CEO and founder of BW Tech Medical, the sponsor of today's broadcast. And today we're looking at light to the brain, photobiomodulation in neuroscience. It's one of the exciting areas where there's a lot of very interesting work being done. Dr. Pryor is very involved in this, and he's going to introduce our guest, somebody else who's been around this field for a very long time. And I'm excited to hear what he has to say and learn a little bit about this very interesting area of current medical science. So, Brian, would you take it away, please?
Thanks, Andy. And thanks, everyone, for joining. We want to keep this light, keep it discussional, and hopefully people can get, some educational points or inspire some questions to ask our guest or Andy or I. We're fortunate today to have Luis De Taboada. Luis is an old friend and coworker. Luis was our Chief Technology Officer at Lightcare, really helped us taking PBM to the musculoskeletal field for pain, inflammation, performance enhancement, and many applications in vet medicine. Luis and I are also co-founders of NeuroThera, which is a company focused on bringing light to the brain for depression, Alzheimer's, Parkinson's, and other disorders. So today, Luis will join us and get some thoughts on brain disorders, how light has an impact, and his history with photobiomodulation and other laser applications. So thanks, Luis, for joining us.
It's my pleasure. Thank you for having me.
First question is, how did you get into lasers? Like, what was your first inspiration? Where were you introduced?
That's best way I can answer that. It was just happened. I had just come to the United States. I was looking to go to college. I had found a job where I was doing some manual work, and it just so happens that the people involved saw some future in me and helped me through my schooling. They were into lasers, so that became my career.
And did you kick right off with medical lasers, or were you involved with other things first?
Yes, so for the first few years that I was doing this, I was an assembler, a technician, learning about lasers, going to school to get a degree in physics and electrical engineering. And my first actual engineered laser was for posterior capsulotomies to treat the eye. This was in the early 1980s, mid 1980s.
Excellent. Yeah, and then roll forward, you've been certainly very influential on many different areas, right? Dentistry, surgery, and photobiomodulation. Tell us how you first got into photobiomodulation.
Well, that was Dr. Jackson Streeter. At the time, I was working for a company called Medical Optics, and we were developing lasers to do dental ablation. So to be able to do cavities or to treat cavities without the drill. And Dr. Streeter was a Navy pilot, lieutenant commander at the time, and he was interested in lasers because, having been to Russia, he realized the Russians were using red light lasers, 632 nanometer Heenies actually at the time, to treat their cervical pain. So he thought this interesting and he wanted to bring it to the US. So much so that he decided to leave the Navy, Dr. Streeter is an MD and a top-down fighter pilot and a friend. And after several months of trying to talk me into it, we went and started Photothera. Photothera initially wanted to focus on cervical pain, but when we looked at venture capital funding, they were not interested in musculoskeletal conditions. So the VC community kind of led us through what they would fund, and the first thing they were interested in was cardiac events, try to treat heart attacks, basically. We engaged the Israeli researcher, Dr. Uri Oran, who again became a friend, and we tried to treat the heart. We did a lot of work trying to treat the heart. We studied the light transmission in pigs. We visited, I personally witnessed several quadruple bypass surgeries. What we found is that the lungs were an impediment to light transmission. So it would be very, very difficult to do non-invasive photobiomodulation of the heart. In the process, Dr. Oran suggested, well, how about the brain? It's a little bit easier, right? There are no lungs in between. The amount of tissue between the skin and the brain is somewhat controlled. It doesn't vary very much between adults. So the thickness of your scalp, the thickness of your skull, we know more or less what those are. And it's not like the difference between a very thin individual and a very heavy individual when you're trying to treat the heart. So that was a control environment. And we spent 4 years developing that technology, doing cadaver studies. The question that comes up, the obvious one, is how can you get light to the brain? The short answer is that after studying it, modeling, doing significant research in the area, we found out that you could. And that led to intellectual property development, multiple patents, because at the time, like everybody else, we didn't know how much light will get to the brain or how much light would be enough to create an effect that would be clinically beneficial.
Yeah, that's great. And then so that led to kind of after the research and transmission and, lots of publications, that I think you guys have done a great job of one, getting a lot of intellectual property with Photothera, as well as publishing a lot of early studies. And then you went into stroke trials. What was the precipitous to go into stroke?
It was an unmet need. So after we demonstrated that we can in fact get light to the human brain, published the results, patented the results, we looked at what would be the most obvious application of that technology based on how we understood it to work biologically. What was the light doing when it got to the brain? With that understanding, we realized that, well, stroke was ideal because the light will allow some of the neuronal and the support cellular structures in the brain to survive hypoxia. So when you have a stroke, an area of your brain basically dies immediately. It has no oxygen. But volumes around that small center have some oxygen, are hypoxic, and then they would go on to die if you don't do something. We recognize that this technology had the possibility of allowing those cells to survive. We investigated that in a mouse model, in a rat model, in rabbit models. So we eventually realized that, yes, this will work. It worked very well in the animal models, but we're also cognizant that the size of the scalp and skull of a mouse or a rat or a rabbit is significantly less than that of a human brain. So when we started our study for stroke, we went with the safest possible dose that we can actually apply to the human brain, knowing that it would not heat the tissues of the brain and that it had a chance to create an effect. Meaning the first in-man application of the technology had to allow for safety considerations because we did not know. Now, our first study, NEST 1, showed great effect. And that started a series of events, funding events, technology development events, publications, further studies that led to another study in which we investigated the same technology on a larger population. So that second study, we found effect, but not statistically significant effect, which, having spent several years developing, meant that we basically stopped. So at that point, we did that technology, and Brian and I started Neurothera. We took the patents and decided, well, stroke is a tough disease. It's a tough problem. Let's try something a little bit more cortical, because some strokes can be limbic, and the amount of light that gets to the limbic system is not enough to help them out. And then we started to do things like depression, Alzheimer's, and Parkinson's. I'm involved in a study to treat autism. So when it comes to treat the brain, we are going to be limited with transcranial, non-invasive therapies to the cortical areas of the brain.
And, it's kind of hard to, when you think about this not being in the field, right? You have this big giant skull to go through, right? It seems counterintuitive that you can get any light through. And I know there's, really good studies and you've done, and we've done a lot of measurements with cadavers and other things. But explain a little bit about how we figured out how light gets through the skull and how it was measured, and that there is actually getting some therapeutic dosage into the brain.
Sure, absolutely. I think that limitation that people have is in this concept of light as being limited to the visible spectrum. So when somebody says light, they think, okay, red light, blue light, green light, right? Well, that's visible light. Outside the light that you can see, that your retinas can perceive, there is a whole spectrum of light that you cannot see, that is not affected by the tissues as much as visible light. For it is true, I mean, if you can get red light through your skull or green light, then you'll need to be transparent to the eye, and we're not. But one way to think about it is in terms of electromagnetic spectrum. I mean, nobody would doubt that X-rays can get through the skull and scalp, right? The X-rays are electromagnetic field that just oscillate much faster than visible light. But when you start to accept that the frequency of oscillation of the electromagnetic spectra determines the penetration of that spectral content to the tissues, then you can start to conceive that, okay, well, maybe there is something that is in between visible light, which does not penetrate sufficiently to create effect, and X-rays, which go through you, or even, cellular radio waves, they penetrate you. So that sweet spot that creates a biological response because it's sufficiently absorbed and penetrates sufficiently through the scalp and skull is near infrared light, somewhere between 800 nanometers and 1200 nanometers. And what the basic idea I'm trying to communicate is, in the electromagnetic spectrum, there's a section of light, near infrared light, that does penetrate sufficiently through these tissues to create a biological response in brain tissue. That said, the penetration is a few percent. So of the irradiance you apply to the skin, 4, 5, 6, 7% will get through on an adult's head. Maybe a little bit more depending on the wavelength and skin color, but it is that order of magnitude, a few percent of the light that's applied to the skin that gets to the brain. The interesting thing is that light gets to the brain. It is quadrillions of photons per square centimeter per second. So that amount of light, it's more than enough to create an effect. And that's what we're working on.
But there's a minimum amount of light, right? I mean, we know that going out in the sun might make us feel better for different reasons, but it's probably not doing anything to help us in the photobiomodulation respect.
Fair enough, yes. There is a minimum amount of light. And not only a minimum amount of light intensity, but a minimum rate at which the light gets to the brain. So even if you have a certain amount of light applied to the skin getting to the brain, you have to do it for long enough with enough intensity. Otherwise, you create some effect that dies out. You create the effect again, it dies out before establishing a minimum threshold of clinical benefit. So we do know that. I mean, Brian, you and I have spent, years now trying to figure out exactly what that number is, right? What is the least amount of light that can get to the brain that would create an effect? Well, the short answer is it depends on the wavelength, it depends on the individual, and depends on the disease state. So Alzheimer's is going to be a little different than depression. When we treat children with autism, significantly less energy because their brains, their skulls and scalps are much thinner. So yes, there's a minimum amount of light. We have a very good idea what the minimum is for an adult when we're treating depression and Alzheimer's.
Yeah, I think that, in a recent study, looking at different doses, right, there has been kind of a dose that seems maybe not optimal because the clinical trials are still going on, but as far as blood flow and other things, can you talk through kind of that study and where it was looked at varied dosing of light and what the outcomes seem to be?
Yes. So we were lucky that we were able to get some researchers with us that are strong in the field. Dr. Paulo Casano, Dr. Dan Iosifescu from MGH and NYU, respectively. And with them, and using our devices, we got some grants from the government, investigate these. There was enough substance to our work that the government decided, NIH, NIA, decided, yes, okay, we need to find out a little bit more. There's just too much noise in the field. People are trying everything and anything and getting results from nothing happens to miracles. There was obviously a range of things happening and we needed to know more. In this study, we investigated 3 different doses of light. Let's call a low intensity, medium intensity, and high intensity light in their effect for cerebral blood flow and depressive symptoms. What we found is that the high intensity light did not have any effect, either in cerebral blood flow or depressive symptoms. Now, there are caveats to these studies being published, and for anybody interested, please contact Andy, we can provide these. But the low intensity light did not modulate cerebral blood flow, although it had some effect on depressive symptoms. It was the mid-range that create the best effect in both cerebral blood flow and depressive symptoms. And we are pursuing that exact dosing in a study in which we're going to investigate that dose for depression and Alzheimer's in a sham-controlled study. Keep an eye on these. We're going to get results in the next 18 months or so, and we will know better. But to your question, Brian, yes, there's a least amount of light that create an effect, and interesting, a most amount of light that stops creating the effect. This is not new to the field. It's been postulated before. Dr. Hamlin had some ideas here, and it's proving to be true. Too much is not good enough. Too little is not good enough. There's a sweet spot, which interestingly, Dr. Gonzalez Lima has studying this technology for cognition in healthy adults, finds that irradiance, that intensity of light that we use is very, very much successful in enhancing cognition in healthy adults. We're talking about 250, 300 milliwatts per square centimeter. The wavelengths are different in the studies. The way you're delivering the light, how much, for how long, at what rate, those 2 data points are interestingly connected.
I mean, the field is pretty complicated, right? Because it's where do you put the light? Is there hair involved? What's the color of skin, right? So I think that it's important for people to recognize that doing controlled studies is critical, right? And I think that depression, because we're treating the forehead, not worrying about hair, has a little bit of maybe more control. Whereas Alzheimer's, Parkinson's, where do you treat and why?
Great question. You have to look at the pathophysiology of the disorder, right? So when you look at things like depression tends to be, or my understanding is executive function, frontal cortex, prefrontal cortex disorder. Now, ideal, right? This is the first place you go if you're going to try to treat with light. You're treating through the forehead. Most of us don't have any hair on the forehead. So, all right, one variable that's eliminated. Scalp and scalp thicknesses vary very little between individuals. Another consideration that's eliminated. Skin color makes a difference, but we understand very well what that is. So when we treat depression, we treat through the forehead. Now, that's all there is to it. Now, that doesn't mean that you're limited to treatments through the forehead. It means that you need to understand how to account for hair. So a simple example would be the scattering in hair, we're not going to be able to avoid. But the absorption in hair is very much greater for dark hair because of melanin. There are some near-infrared wavelengths that are minimally affected by absorption of melanin. So if you really want to approach treating through the hair, you use something close to 1200 nanometers, 1170 nanometers. That limits that effect. If you put it all together, it's more than just absorption, right? The light is absorbed by chromophores in the skin, in the hair. But what people have to understand that it's missing in the overall community is that the driving mechanism for light energy losses is the scattering. Scattering of tissues is 10, 20, 30 times greater than absorption. So this is one of the things that I would encourage people listening is pay attention to what's happening to the physics here. Absorption is one factor. It's not even the biggest factor for light transport through tissue. When people say 810 nanometers is the best wavelength to treat, maybe it's the best wavelength for efficacy, but it is not the best wavelength for transmission. You can easily model, demonstrate, and it's being measured that you would do much better using, in some cases, 980 for dark skin color without hair. In some cases, 1064. 1170 is being used by a company in England. If you are considering only transmission through the tissues. So,
I think it's important.
Complicated, we know how to do it.
Luis, just let me ask you one thing while you're on that particular subject. Is pulsing versus continuous wave, does that impact how light is transmitted or impacts efficacy in any way?
It does not impact how the light is transmitted. So the peak irradiance during the pulse, if you think of a pulsed light as being something that's being turned on and turned off, the intensity of the light during the on time, it's the same as one that's applied continuously, then there is zero impact to the penetration of that light through the tissue. During the on time, you get the same amount of light at any depth as you would with continuously applied light. Now, if the irradiance during the pulse is much higher than you would use during continuously applied light, for that short period of time, the irradiance at depth will be proportionally higher. No magic here. The only thing that it's really different, if you in fact determine your pulsing rate and duty cycle properly, it's that you can get, for short periods of time, more light through the tissue without heating. That's it. So for the biophysics of light transport, pulsing does not make a difference for light penetration. Now, for efficacy, that's a different question. Short answer is we don't know. We have seen things like, I did a study on Alzheimer's in mice, and what I was able to determine was that CW light worked, but it didn't work quite as well as 100 hertz, 20% duty cycle light. Why? It could have been thermal. It could have been, there are too many variables to consider. So for pulsing, people tend to think, okay, there's something else going on here, and it's likely to be the case. But today, we use CW light because it has the most experimental data, and we have seen with it the best cerebral blood flow changes. So short answer, yes, pulsing is like to make a difference. Long answer is we don't know exactly how, and we don't know what the best parameters are. So this is a whole exploration space that's going to take time.
Luis, maybe explain to the audience, how do you measure blood flow? Like how do you know that you're improving blood flow into the brain?
Sure. So the way we did this particular study is using MRIs. So there are techniques, without getting too deep into these, that will allow you to measure the oxygen level concentrations on the blood in the brain. Now, when you do this indirect measurement, if the oxygen levels increase, the underlying presumption of the technology is that, in fact, there is more blood flowing. It makes sense, right? Blood is carrying oxygen from the lungs all the way through your body to your brain. If you can say that there is a higher concentration of oxygen in the cerebral structure, there is higher blood flow. That's the basic understanding. So in our study, what we did is we said that exactly. We're going, okay, put a person in the MRI. We took pains to design a device that would bring the light into the patient while inside the MRI. So as you might imagine, everything in this delivery system had to be non-magnetic. We tested it, we developed the technology, and then for each patient, we investigated what happened with low, medium, and high intensity light. So, and control, so a sham environment. So each patient was his own control, and we were able to tell for these patients, well, what happens? How much oxygen is flowing through their brains? And the medium low dose of light had the best effect. Brian, we're doing a study in Peru where we're investigating a flip side of these, which is the metabolic state of the cells of the brain. So we're using PET, positron emission tomography, to see if the cells are using the oxygen beneficially. So they're spending more energy. They're actually creating effects in the brain to survive, to reduce inflammation. We will have these results in the next 6 months. And at the same time, we're correlating PET results to EEG results and to depression scales, MADRS and others. This study in Peru is going to give us a connection between the dosing in which we found cerebral blood flow changes. We're using the same dosing in Lima, the 3, medium, low, high, but we're investigating that for cerebral metabolism, electroencephalography. Are we changing the way the brain patterns are happening in their frequency domains, and correlating those to depression scales? A subjective thing is the patient feeling better. So I think in the next 6 months to a year, we're going to have a very clear picture of what's going on for depression.
Yeah, I think it's critical, right, that as fundamental as measuring the light in a joint or in the brain, or, even doing, you've done a lot of modeling in the past and published, making sure that it makes sense, right? That you're putting light on the tissue. It's not heroic, right? It's not easy, but it's not heroic to do these measurements in a lab to make sure you're collecting some photons where you say that the tissue is going to have a response. And I think one thing that you and I strongly agree on, which Andy's work for us, we do disagree on many things, but we strongly agree on biomarkers, clinical outcomes. That's why we're taking these heroic steps, not just going to market and selling something, right? We want to make sure that we have some kind of evidence that we're getting light there first, that we could explain what's happening, and then the most important, that we're seeing clinically, evidence versus a sham.
Yes, I absolutely agree. I mean, I believe it was Carl Sagan that said extraordinary claims require extraordinary proof. So one of the detrimental things in our field is that there are so many light sources, LEDs, different wavelengths, different frequencies, different intensities. And it is, like you say, a rather easy thing. Build yourself, get a flashlight, and put it on your head and see what happens. And it happens everywhere. So when you have these enormous amount of variables for a large number of disorders, Parkinson's, Alzheimer's, depression, autism, with people well-intended trying to get some effect. What you find is, yeah, well, somebody publishes something that isn't, it's a miracle, right? A case study of 3 patients that just incredible. You have another study that failed. And people tend to put it all together under PBM saying, well, this is kind of voodoo. What's going on here? There clearly is a signal. So for us, when we talked about these, we said, okay, objective measurements correlated to subjective outcomes. In order to get FDA clearance for our devices, you have to meet their criteria. So they say, okay, if you're going to treat depression, you have to measure it this way, whatever that is. Usually subjective scale, MADRA. But nobody buys it. I mean, it's like, what are you doing? There is so much the placebo effect. When Brian and I talk about these, okay, let's measure PET. Let's measure cerebral blood flow. Let's measure electroencephalographic changes. Those are objective things, right? It'd be hard to say you're faking cerebral blood flow changes, especially when you have a sham, and the sham is the same person in which you tested things. And PET, metabolic responses, electroencephalographic responses. So we're putting these together, and I think the field is going in the direction of doing some serum whole blood measurements of inflammatory cytokines. You know, when you start to think about these, and you probably read, most of the audience that are listening, chronic inflammation tends to be a background problem for a lot of disorders. Mitochondrial dysfunction leads to chronic inflammation. Applying light to the mitochondria increases or restores their normal function. Then inflammation should be reduced. That's an easy thing to measure, right? Let's find out which cytokines. Interleukin 6, interleukin 10, BDNF, we can measure those. But there's no established baseline. So we're starting with imaging techniques, PET, MRI, EEG. But we are going in the direction of having some tests that you probably can do at home, that you'll be able to say, hey, I'm trying these and it's working. And ultimately, I envision these to be a personalized therapy, a device that is easy to wear, you can take home, you can measure in saliva effects to your chronic inflammatory cytokines, and you can adjust it or adjust it via a closed loop with some smart server that is using ChatGPT, some generalized AI, to determine how these things are moving. Like I said earlier, there's too many variables, individual variations. Eventually, we're going to find something that works. We're very close. And then we're going to use that as a starting point to allow people to individualize their therapeutic parameters in some safe way.
Yeah, the future's definitely very, very exciting in light therapy, and there's enough for all of us to do. What do you see? I think you gave us a little hint of where you think the technology is going, but what do you see kind of big picture? What are you excited about in the world of lasers in medicine?
In the last year or so, I'm excited about the acceptance. With the negative side of everybody using it, however, there's a positive that says they are trying things, and by far, most of these are safe. One of the great benefits of transcranial PBM is that there are no known side effects. You know, there's little signals of effect, right? People are trying 1170 or 1070 nanometer wavelengths, and they're trying it with a cast. They're trying it out there, and they're getting some response. So all these things contributing to our knowledge of what's happening. And in the future, what I see, there's going to be a coalescence of wavelengths being used, irradiances being used, and it's going to be divided into a wellness space with the low irradiance device is going to have some effect, emotional responses, because light gets through to the brain. And the clinical, more substantive effects in clinically approved devices for depression, Parkinson's, and Alzheimer's It's going to have a higher irradiance. It's going to be more substantive in their engineering. It's going to require additional safety features. So it's going to split. It's going to split into two spaces. Wellness devices, a lot of this is being tested right now with some interesting effects, and therapeutic devices, which are going to have to go through market clearance and all the safety testing that are required for FDA and CE. It's coming, and we're understanding more and more what's going on and what it takes to make it work. I can see the next year and a half or so, devices out there to treat depression that are clear for marketing by the FDA. Shortly thereafter, Parkinson's is looking very good. We have just finished a study in Spain. We got beautiful signals of effect, feasibility. Alzheimer's would be a holy grail of this technology, but that is probably going to take longer as we try to understand the pathology and what's going on with the disorder, the disease.
Luis, that's great. As we wrap up here, you're very much an evidence-based person, as we've heard today, but sometimes evidence of your own eyes. Is there a case that you would share with us where you've seen something perhaps miraculous or something that really encouraged you that you'd like to share with our audience?
Oh, many. I'm sure Brian can say to these too, but one of the most rewarding things of being a nerd in this space, and I'm not an MD, is exactly those cases where it's just a miracle happens. Our third case for stroke was an 82-year-old grandmother. And to treat stroke, we had to shave the head, which was a limitation of the technology. We talked about this before, hair. And as a physicist, I'm going, I don't need this variable. I do not have to deal with this. If you want to be treated, your head's going to be shaven. And we did. The lady, slow grandmother, was hemiplegic. So the right side of her body was paralyzed. And it had just happened within 4 or 5 hours, if I remember the case. Jackson and I were in Lima, Peru, treating these patients. And when she came in, the doctor said, you guys are the designers. Dr. Streeter, you're an MD. We did things the right way. We went in there and tried to help the lady. Within 40 minutes of applying the light, her speech returned. Her right side paralysis was resolved. Of course, the skeptic would say that would have happened anyway, right?
Sure.
But we saw sufficient of those cases against control cases because the control cases were not resolving the same way. So exactly how it happened, the area, the location of that stroke had huge impact on how they resolved. But
I can see how that patient is stuck with you and encourage you to carry on, right? That was great. Thanks for sharing that. I appreciate it. Brian, do you want to just Brian, you've got a foot in commerce and science. Do you want to just give us an overview of where you think this may be going and what you see for the future from what Luis has shared with us today?
Yeah, I think good science, with good published clinical trials is going to raise the bar, right? I think we're seeing now we're narrowing in on, parameters that we're going to see some significant results. And I think it's going to take, that one application to get through the FDA. I mean, let's face it, even once we get FDA approval and get to market, it takes 10 to 15 years to become standard of care, even things that work miraculously, right? And so I think it's going to take a long time to be standard of care. I'm very encouraged by transmagnet cranial therapy, right? TMS has really plowed ahead. I think a nice pathway for light therapy. It is, energy on the brain. There is approvals for depression, for OCD, for many things. Insurance reimburses it. So I think there's a pathway that we can follow that we're not kind of the first technology to be a device on the brain. And so I think once the science is there, I think there is a clear pathway, and I think it's pretty exciting to explore some of these other diseases that don't have anything right now.
Yeah, this is a great start to laser pulse because really, it's just cutting edge and looking forward to dealing with some of these things for which there seems to be very little options, particularly in depression and Alzheimer's. Luis, any closing thoughts from you?
Closing thoughts for your audience. Pay attention to the physics, pay attention to the mechanism, pay attention to the underlying events that photobiomodulation is eliciting in tissue. Just trying anything is great, but your chance of success on whatever your endeavor is are much greater if you can actually assign some reality, some double checking to what's going on. I see too many therapeutic wellness devices that are trying things for the sake of trying things. There's a lot of science. Look it up.
Yeah, there's a lot of science. Look it up. There's a great note to go out on. I'd like to formally thank you, Luis, for your time today and sharing what I think has been very insightful and very helpful. I'd like to thank BW Tech Medical for sponsoring this. If you have any questions for Luis or any questions about laser in general, info@aimla, AIMLA, info@aimla.org. For any comments, negative, positive, we welcome a dialogue and look forward to our next episode, which will cover another area of light in medicine. Thanks very much for attending.
Thanks, Luis.
Thank you, Ryan. Thanks, Andy. Take care.